Let's look at the next question, where you have to identify the true statement. And the first one is the process of spermatogenesis and oogenesis is exactly similar. Now this is false, and as I showed you in the previous in that image, and let's go back. And as I showed you here, notice that these four spermatids, they form four sperms, and this process goes on till the sperms are formed. Whereas this is one main difference. Whereas in oogenesis, notice the secondary secondary oocyte will form an ovum only if it is fertilized. So if it's not fertilized, the process is not completed. So that's another difference. So here, and the second difference is that there are four functional cells. Here we have only one functional cell, and we have three polar bodies which are non-functional, because we want one cell to have all the cytoplasm, and also we don't want a population explosion. So these are some two major differences which occurs. And the other thing is that oogenesis begins before birth, whereas spermatogenesis does not. It begins at puberty. So you can see that there are quite a few differences, so they're not exactly similar. So here, this is false. This sperms attain complete motility in the female genital tract. This is true. Sperms attain a great degree of motility in the epididymis, but only when they are deposited in the female genital tract do they attain complete motility. So this is true. But let's look at the other one. Identical twins are formed when two sperms fertilize a secondary oocyte. This is false, because identical twins are formed when one sperm fertilizes a secondary oocyte, and then the zygote divides into two, and each part then forms an individual. Fraternal twins are formed when two sperms fertilize two secondary oocytes. Now in this case, when two sperms fertilize one secondary oocyte, you will have more than the normal number of chromosomes, because you'll have 23 here for the sperm, another sperm 23, and that for the oocyte. So you get 69 chromosomes, which is, then life is not possible. So actually you get a non-functional zygote, and the zygote just dies. So this is false. The fourth statement, which is amniocentesis is a procedure where you look at fetal urine. This is also false. In amniocentesis, what you're looking at is the fetal cells, the cells which are from the skin, which get sort of abraded and are deposited in the amniotic fluid, and you're really looking at the fetal cells to look at chromosomal abnormalities. And number 5, the son inherits X-linked diseases from the father. This is also false, because the son is a male and he has XY chromosomes, the sex chromosomes. The Y comes from the father, the X comes from the mother. So therefore, X-linked diseases, the son always inherits from the mother, not from the father. So therefore, in this, the answer to this statement is, this is the true, this one here is the true statement. Let's look at this question, which is a clinical problem which tells you about height, and it gives you a situation where T, which is uppercase T, is for tall and is a dominant gene over lowercase t, which is short. Then it tells you that 50% of the offspring in any pregnancy will be short. So they will be short. It tells you that. And then asks you what is the genotype of the parents. So here you're working backwards. You're given what the result will be and asked to find out what is the genotype of the parents. So let us look at, let me go back to red color, and let's make a Punnett square and work with the information that we already have. So we know that the offspring are going to be short, which is they will be like this, and 50% are short. So out of 4 possible combinations, 2 will be this way. So I'm going to write the 2 in this part here. We could write it anyway, so we can write it this way. Now, obviously both parents cannot be homozygous short, because if both parents were short, so that means the father and mother were like this, we would have all 100% would be short. So both parents can't be that. So we can erase this. So let's erase this. And the same thing also goes for both parents cannot be homozygous tall, because then all offsprings would be tall. Because if you had all capital T's, we wouldn't end up with a lowercase t at all. So let's work with, say, both parents are heterozygous. So they have one tall gene and one short gene. So they have one tall gene and one short gene. And let's see what the combinations will be. So this would be this way. You work, this would be this way. Now this would be this way, right? So then only 25% would be short. So this doesn't fit the, so one parent being or both parents being heterozygous also does not work. So we've discounted this one and we said they can't be this, and then both parents being heterozygous is also not doesn't work out. So again, let me go back to let's work out what the possibilities can be. And here, and so we come back to this, and okay, let me erase this one. Now let's say we have one parent who is tall and one parent, because that's what we are left with, and one parent who is homozygous and short. Now then you will find that these are the possibilities you will get. This way, and this will also be a capital T, and this will also. So all children will be tall. So even this does not work out. So therefore this is also not does not work out. So we can see that this is most likely the right answer, and let's work this and see if we can get that. So I'm going to erase this, erase this, and erase this. So we had that one parent is heterozygous tall and one parent is short. And let's see if we get 50% as short. So we get this way. This and this works out this way. This and this will be a capital T and a little t. So I can erase this. And this is little t. So we can see here we do have 50%. I just had it in the wrong place, but it's okay. We do have 50% who are going to be short and 50% who are going to be tall, but they are going to be heterozygous. So therefore the answer is this one. So this is how you're going to work out backwards when you need to find out what is the genotype.